Good day. Welcome to the Waters and Tea Instruments Medical Device series. This series aims to show you how we can help you understand your medical devices better from a chemical materials characterization perspective. This can then be applied to product development, to processing, to regulations, to troubleshooting, presented each week by an expert in the respective technology. For this webinar, we encourage questions, and we'll have some experts on hand to answer as many as we can in the time we have. Those that we can't get to today, we'll follow with afterwards. Please post questions in the top left window and we'll answer at the end of the webinar. There are links to resources in the bottom window and a survey in the top right, which we'd appreciate you completing. I would also invite you to register for future episodes of this series. This series will comprise of multiple sessions, one a week. These will cover various aspects of medical devices and the materials used for these devices. And we'll highlight how some of the technology Waters and Tea Instruments have can answer some of the important questions that you have. Today's talk is the mechanics and strength of biomaterials. I'm Ben McGreath, responsible for the medical device market at Waters, and will be your host today and for each session of the series. Today's presenter will be Troy Nickel. Troy is extremely experienced with the Electroforce product line, being the technical lead for over 20 years, and is very knowledgeable in the applications that it serves. Troy enjoys bringing his depth of knowledge to enable our customers, helping optimize testing and research so they can solve the challenges that they have and push their research forwards. Within the broad world of medical devices, last week we spoke about chemical characterization and some of the regulatory changes that have happened of late. This mainly focuses on the small molecule moieties that can leach from the device to a patient. This week is very much focused on mechanics and the strength of the biomaterials and the tissue that will be replaced or that will surround a device. Troy.
Hello. Today I'll be presenting to you on tissue and biomaterials, and specifically mechanical testing, and covering examples of mechanical tests that are used with our instruments to characterize materials, oftentimes in the context of future design of medical devices. So let's talk quickly about the difference between tissue and biomaterials. Sometimes biomaterials, they lump tissues into there, but today we'll keep them separate in that tissue is going to be native tissue, so biologic tissue. Most of the context is of human origin in terms of medical devices that we're designing for humans, but we'll also see animal tissue used as models in research. So these types of mechanically loaded materials that we're usually working with are things like bone, ligaments, cartilage, some heart valves is a very active area right now. Where on the biomaterial side, these are going to be synthetic materials designed by materials designers and biologists, and mainly used to restore function of a body tissue, often a diseased tissue or a failed tissue, or maybe to augment a tissue that's there. So here examples might be ceramics used in dental areas, ligaments, but synthetic ligaments, synthetic vessels, tissue engineered cartilage, and heart valve components. So in this context of medical devices, from the tissue side, it's important to understand either tissue that's being replaced, to understand its properties, to replace it with a material of similar properties, and on the biomaterial side, to understand its properties to compare to them that native tissue, or in some cases, they're native tissue that are going to be surrounding the man-made material, so it's important to characterize those materials. I'm actually going to turn off my video going forward so you can see the entire slide. So here's an illustration that we like to reference in terms of laying out the development pathway of maybe a medical device, implantable medical device, where in the forefront is going to be a completed device and you're going to be doing final tests on that. But oftentimes years prior, or maybe even accessed in research publications, you're looking at the native tissue, and a medical device might replace that tissue, or there's tissue surrounding it, and it's important to understand the characteristics of that tissue. So that's kind of a first step that we say on this pathway. But then as you go forward here, you have to characterize, maybe there's biomaterials that you've designed that you're going to assemble into a device, or you're going to use some native tissue maybe decellularized native tissue, and you need to characterize that tissue. And then as you go forward, you're often looking at more of the performance in terms of durability, reliability. This is an area we do quite a bit of testing in, and that could be of the materials themselves or subcomponents of those materials. And in this case, the context here is a heart valve device where you might have metal or a superalloy wire. You might have biologic tissue that is the heart valve leaflet that's going to get sutured onto those. So every one of those individual components needs assessing, as well as the assembled component. And then finally, there could be fatigue tests and performance tests on the complete devices. And again, all those in the forefront are going to be generally according to some standards, some industry standards, and that gets less and less as you go back in time through this pathway. So hopefully this illustration is helpful. So today we're really going to focus a lot on the native tissue characterization and biomaterials and tissue that are going to go into the devices. So we're going to really cover those first two steps on this pathway. Today we're going to highlight a few applications. We're going to have 6 applications total, and 2 in each section that we divide out this area. First is the tissue characterization, so native tissue characterization, then biomaterials, and then finally some examples on tissue stimulation, often used in regenerative medicine or tissue engineering. Okay, so the first area, tissue characterization, we're going to look at dentin and we're going to look at bone. So this first study is I think a nice foundational study to start with, because it talks about a couple different paradigms of testing types and how they play out in assessing strength. So this is on human dentin. So dentin is the center part of your tooth, the material in the inside. And the purpose of this study, this was a University of Maryland study, that they look at the strength as a function of age, and looking at 2 different characterization. First is ultimate strength, and then is fatigue strength. And I'll talk about the difference there. I like how this study highlights both of them. The samples here are cut out of the center of the tooth. So the dentin is not the enamel on the outside, but it's the inside. And it's going to be repetitively loaded in what we call 4-point bend. So this illustration on the right, where you've got these pins pushing on the top of a bar, and that bar has pins on the outside. So as you apply force from the top, you create a curvature. That curvature creates stress and strain in the material, and it's we can characterize that nicely to understand strain and stress and where the material fails. So in this case, there's this small, really miniature fixture. As you can imagine, these teeth are very small. So 6 millimeters in what they call span, the bottom part of the sample points, and the top part is 2 millimeters apart. So that miniature fixture designed at the University of Maryland inside of this instrument, electroforce instrument of ours, that is a 450 Newton capacity instrument. Now, we might use much less force to fail the material than 450, but that's an attribute of the instrument, as we can use it over many different force ranges. So here we see a little bit more information on the samples. So in molar samples, we've got this section in the center. That's what we're taking these 6 millimeter length samples out of. And that, if you look at it closely, the micrographs show that there's actually what's called dentin tubules. Tubules are what's going to allow nutrients to pass into and through the tooth. And as we age, they become calcified. So we see calcification here. We obviously hear and read about calcification in vessels in terms of heart disease. Well, here we actually see calcification through these pathways of nutrient flow. So as you can expect, that's going to impact that material over time. And on the right, you see some of the results as you plot the strength. So the maximum bend strength stress to failure on the top plot is on the y-axis, and that against age you can see is significantly reduced, pretty much cut in half by the time you are 50 years old, and even less as you get older. And energy to fracture, which is sort of its capacity to resist loading. Another way to measure it also is reduced significantly. In this case, even more than half reduced by the time you're 50 or 80. So this is one way to look at these results. But if we sort of back up a little bit, we'll see. So here we see a little bit more of the illustration of the data, the different types here, so that those plots we just saw would have come from the first plots on the left. This is what we call a monotonic, means one direction loading, constant rate loading, where we're applying bending to this 4-point sample until it fails. And when we do that, we're applying strain, but we're measuring a stress, and that stress goes up and up and up. And that first section, that first linear section there, you can see denoted E1 and E2. That slope is essentially the modulus of the material. And here we see that the modulus of the material is in the first section, but then as you increase the loading, the young tooth starts to sort of continue, yields a little bit, and it goes to almost over 100 to 150 megapascals. But the older tooth down below, you'll see there's a couple examples there, 2 samples run. And interestingly, the modulus isn't significantly different between these. So it's not like it's harder. It's that it has less capacity to withstand the additional stress. So that area under the curve is that energy we talked about in the previous plot, and you can see that ability to resist that energy is much reduced in the older teeth. So that's the type of data you get when you have a single load to failure. But as you repetitively load it, now you get plots that look something to the right. So this plot, let me unfold this a little for you. In the top right, there's stress amplitude. So how much oscillating or repetitive load are we applying? And then for that amount of stress, how long does it last in terms of the number of cycles? And the lower plot is essentially just a fit of that data. So here you have lots of different samples, and those samples, depending on, every little sample is going to be a different, you're going to get scatter in terms of testing multiple samples and what their results are. So you can see the scatter of the young group is on top in open circles, and the scatter of the older group is in the darker circles. And now you see a significant reduction in not only the strength, and as we saw on the left, but we see that played out over time in terms of oscillating load as well. So for example, if you said for 50 megapascals, so if you imagined a line from 50 and you draw that over, you see that the older samples will last something like 1,000 to 10,000 cycles of repetitive loading. But if you take that and draw further over to the cluster or the line in the young data, now you'll see that will last almost 1 million cycles. So now you see that length of time is significantly longer for the same amount of stress. And as these teeth get older, they become much less resistant to repetitive loading. So in conclusion, I think this is just a nice study in terms of, okay, there's a variable we're looking at, and that variable is age, and we can look at it a few different ways. We can look at it with monotonic loading, so single push to failure in this case, and we can also look at its resistance to fatigue loading. And these are illustrated nicely by these 2 sets of plots. Okay, our next study, we're gonna now focus on bone, and I'm going to introduce this unique study type called in vivo loading. So this is about applying load or force or stress or strain, you can think of, to bone, but you're doing it inside of an animal model, specifically a live animal model. So this study is from Notre Dame, and it's a nice study because it shows a lot about the elements of assessing how much strain is on bone and looking at how that strain, when repeated over time, can impact how bone is formed. So it allows researchers to really understand the natural processes and how those bone reformation or formation or bone loss, it can be quite a function of the mechanical load that bone is subjected to. So the instruments used in this study are actually two different instruments. One's the lower force instrument, a 200 Newton instrument, and the one on the right is a 3,000 Newton instrument. Okay, so there's quite a bit to unpack here. So I'm going to work from the left to the right a bit. So here, the study is on the ulna bone, which is one of the two forearm bones in this animal model. And the notion here is we're using these highly precise, highly accurate loading instruments to compress slightly the ulnar bone, and that's going to create stress and it's going to create strain. And the researchers then can look at how does the bone react to that over time in the animal model. And in order to understand that well, we see here some really nice, thorough images and characterization of how much strain do we actually get and where is that strain in the ulna bone as a function of force. So the plots on the right are essentially a calibration curve of sorts where they look at, for a given amount of axial force applied to the joint, to that forearm, in the ulna bone, you've got compression stress, or strain in this case, and tension strain. And looking at over multiple animals, characterizing then how much strain do you get when you apply a given amount of force. So what you'll see later is they then vary the force for different specimens and then look at how that bone is formed at those various levels of force. And because we have calibration curves, we can then understand how much strain was actually on that bone for that specimen. In this case, there's a subtle difference between static load calibration and dynamic load, which is a nice thorough investigation because what you'll see is the loading that they'll actually apply as a dynamic load. So it's important to understand the strain calibrated in that environment. Okay, so a few more details here. So now looking at the bone and the strain, looking at it for 100 newtons of axial compression, now you can see the bone, this sort of illustration of the bone on the top plot. The blue illustration is going to be the top section of the bone. It's the cranial bone in the animal versus the cranial side versus the caudal side. And now you get a profile of strain sort of throughout the length of that. So I guess the high level point is this is a nice thorough investigation of where is the strain occurring and how much is the strain occurring for a given amount of force. Okay, so now as we play this forward and you talk about the loading scheme here, the loading scheme is to apply 2 hertz of compression for only 360 cycles a day. Okay, so that's only 180 seconds total with one of the precise Electroforce 3200s, and then vary the levels of that depending on the specimen. So this is somewhere between 60 newtons to 120 newtons. And just to note, this 3,000 to 5,000 isn't an extreme amount of strain. I think the paper references that typical sort of moving around, running around is in this sort of 3,000 to 4,000 range. So it's not an extreme amount of loading. They're not failing these ulnar bones. They're just precisely applying force so that they can then more precisely understand the consequence of that force. Okay, so getting a little more detailed here is you can see now that they're adding markers at different points of this study. So the study is approximately it's a 35-day study. And depending on the sample, okay, so because they're marking it, we can now see how bone is being formed. And the images on the right are really neat images. They're a little bit hard to see, but you can look at different types of bone and how they're formed. And in this case, on the top, it's relatively low strain, and on the bottom, it's much higher strain. And the table is essentially showing us here that less than 3,000 microstrain, we get very little bone formation. And now we get a little bit more at 3,500, and then at over 5,000, we see significant woven bone formation. So this is just a kind of a neat study on looking at the amount and the different types of formation of bone as a function of strain. And this study is just really nice and thorough on understanding how much strain is occurring at which part of the bone, and then sectioning it at that part of the bone to then look at this reaction and growth and remodeling as a function of the force. Okay, so now we've covered a couple examples on native tissue characterization. So we're going to switch gears now and talk about biomaterials, or synthetic man-made materials, with a couple examples. So in this first study, we're going to talk about hydrogels, and specifically talk about some work through and from Johns Hopkins University. And hydrogels are interesting in that they are sort of hydrophilic by nature. The polymer, the synthetic biomaterials, there's some strong ability to control what the mechanical properties of the hydrogel is, but they're not always bioactive. But biologic materials, obviously, they are proven to proliferate cells, and this study is really around how to really get best of both worlds. So the purpose here is to characterize mechanical properties. And what I think is interesting here is this synthetic biomaterial can be tuned or designed as a part of the formulation and the processing to provide different mechanical properties. And we can see how they're going to measure these properties in the next few slides. So this is chondroitin sulfate, polyethylene glycol. It's a common hydrogel, and it has properties to be an adhesive too. So you'll see there's a part of the study is around adhesion. So we'll talk a little bit about the specimens here. There are cylindrical samples, almost disc samples here, 10 millimeters high, 8 millimeters in diameter. And you can see the kind of different variables here, different ratios of CS and PEG, different relative humidity during the synthesis, and then also varying the concentrations and molecular weight of the hyaluronic acid. So those are the sample groups. From a methodology perspective, they're using one of our ElectroForce 3200 instruments, 200 Newton capacity, and they're going to measure a compression force by applying a compression strain, so like a increasing strain from 0 to 10% strain. You can see at this strain rate. And they're doing this between platens. Some people call those parallel plates. And a little bit of an element here that's interesting is a 2.5 Newton force sensor. So in an instrument like this is a 200 Newton capacity instrument, but if this material will only yield, let's say, 1 Newton or less than 1 Newton of force when you do this compression, it's helpful sometimes to put a lower capacity force sensor. Basically boosts the signal, gives you more accuracy, more resolution in measuring the compression force with more accuracy and more precision. So here's one of their first plots with the resulting tangent modulus and a couple different variables here. The x-axis is varying the pH of the precursor solution, and then secondly, in the different bars, 20% or 60% is the relative humidity change during the processing. So let's talk about tangent modulus just for a bit. essentially you can think of this as stiffness or hardness of the material, but specifically the modulus is the slope of the line between stress and strain. So I'm actually going to pull back the example for from the dental application, where here the stress versus strain produced, this modulus, a line that looks something like this, although that should be straight. A straight line fit here, but in many of these materials, it's nonlinear. In the dental application, it's nonlinear because of yield. But if we were going to say, let's measure the tangent modulus at, let's say 10, in this case, 1% strain, then it would be the tangent of the force stress versus strain plot here and here. So those would be the two tangent modulus for these two samples. So that's how tangent modulus, sometimes you'll see that and be like, what's the difference between Young's modulus and tangent modulus? Young's modulus will be this sort of classic linear line in relatively linear samples that you're fitting here, and tangent modulus will often be used for nonlinear samples. So okay, so that's what modulus is, but let's look at this variation. So now we see a significant variation that as you increase or decrease the pH, we get higher tangent modulus. So that's a stiffer, harder hydrogel. And then secondly, you can significantly vary the relative humidity during processes. So you can think of these as like knobs to turn. When you're making these materials, you can dial these knobs to get the modulus as you would like. And as we talked in the native tissue characterization portion of this, understanding what that native tissue is that you might replace is helpful to then dial these knobs to the right levels. Okay, here's a couple other results. This is a little bit more complicated yet. So before we just did a compression sort of ramp in displacement, which is strain, and measured the force, which is stress. And now we're actually, they did an oscillating study. And this oscillating study allows you to oscillate displacement on this material, same material, compression cylinder or disc, but now you can measure the modulus by looking at the amplitude of force compared to the amplitude of displacement, or specifically the amplitude of stress divided by the amplitude of strain. And that's a method to get modulus, Storrs modulus. What's interesting in this additional analysis is you can look at the damping. So this damping term here in tan delta is now a measure of the force plot, that sine wave of force, and the sine wave of displacement. And any phase delay in a what's called a DMA assessment or a viscoelastic assessment is now going to show you the viscous portion. So as that delay gets more and more, the delta or the tangent delta gets more and more, it means it's a more viscous material versus if it's very little damping, it'd be more like a spring, have very little viscous to it. So as you can imagine, in a case, maybe cartilage is a really good example where you've got cartilage in a joint and you're walking and you're running, you not only need the modulus to be right, but you also need the damping to be right so that all these different frequencies of loading, that the resulting force that material is going to see is relatively normal or similar to the native tissue. So, sorry, that's a long explanation, but the point is that's why you look at tan delta, and by looking at tan delta, you can then try to match that property. And what you see here in the x-axis, as they change the ratios of constituents there, you can significantly affect the damping. But if you look on the left, you don't change the modulus very much. So you now have a knob to turn that is specifically driving damping. So you have other knobs that we turned on the previous slide, which is to get the modulus right, and now there's another knob to turn to get the damping right. So I thought this was a very interesting sort of multivariable study with multiple ways to analyze this data to show these different parameters. Okay, and then finally, this is kind of a simple plot, but shows significantly better adhesive strength. And this is really the goal, the property of this, the goal of this hydrogel development was to create a better adhesive. And that adhesive now shows an order of magnitude increase in adhesive strength. So this is done by what's called a lap shear test, where you have one material. I think they used cartilage on the top and cartilage on the bottom, and then you overlap them. So you make a lap shear test, and you bond those two materials together with this adhesive. And then you pull them in the shear direction, in this case up and down, and you measure the maximum force. You can convert that to stress, and you look at that adhesive strength. So that's how that test is done. On the same instrument, similar type of force, probably more forces than we did with that low force load cell, but to measure in this case, the final result. Having tuned all those other properties to then get the primary advantage here, to use some of the additional properties of this biologic material combined with the hydrogel to create better bonding in this tissue to create a better adhesive joint. Okay, so now we'll switch gears to the eye, and we'll look at lubrication. So this is a study through the University of Calgary, Canada, and their collaborators, where they're looking at the lubricating ability of essentially like a saline solution that you would have with a contact lens wearer, but in this case enhanced in working on the concept to say, hey, can we use proteoglycans, which are found in joints that reduce friction in joints, to add that to this a solution and essentially help people who wear contact lenses reduce friction in those contact lenses, reduce irritation, and have a better experience. So in this study, the resected cornea, you can see in the bottom image there, is actually attached into the lower portion of, actually the upper portion of the instrument. This is a 3200 instrument, kind of like we've seen before, but this one's a little special in that it has a rotational motor, that black part in the middle, and that rotational motor allows us to rotate the lower fixture. So in this case, that lower fixture has the cornea on it. The top actually has the lens on it, and as we rotate that, then we can create shear. And with that PDMS disc there, you can essentially create shear using that lens material. And then we can look at the sliding friction. As we apply force from the top and rotate from the bottom, we can now measure a friction quantity. We'll talk about that in the next slide. And that can allow these researchers to understand, with these various combinations of materials, which ones have better lubrication qualities. So on the test method side, we see this combination of that linear motor on the top is programmed to do different forces. So that's like normal force in a friction equation. And then you're spinning that rotational motor below at different rates, and you can convert those to millimeters per second. And you can see this sequence of events, and the software allows you to sequence this both with sliding velocity as a variable and axial or normal force as a variable. So you can kind of do this all in one kind of test experiment. And you can see the plots later will allow us then to pick apart these different variables. So that's how the test method is executed on this instrument. And from the different samples and materials, we've got saline, we've got different doses of proteoglycan 4, and then looking at a couple combinations of, is there a synergistic effect between this hyaluronic acid with proteoglycan 4 in saline and in aquafy solution. So those are some of the change in variables on the sample side. All right, so now we're going to get kind of lots of data here. But at a high level, I'll try to pick apart the main points. So on the left, we see, material in saline. And then on the right, we look at this dose dependency of this proteoglycan 4. So we see that the sliding velocities on the x-axis, and then static friction and kinetic friction are what we're seeing here. So as you know, as you remember from physics class, static friction is the resistance of motion from 0. So as you start to move, you get an increase in that shear force or torque that we're measuring in the instrument. But then as it continues to slide, then that kinetic friction starts to come into play and creates less force. So let me walk you a little bit from left to right. So on the left plots, we've got static friction and kinetic friction, and there the different curves are the number of times that the shearing is happening. So the first time we get a low number, and that's good, you get low friction, but then as you pass by that same material again, you repeat that motion, the friction goes up and up and up, the static friction. The dynamic friction also goes up, and a little less bunched, but also goes up. So this is, you can see sliding velocities again on the x-axis, and then the first pass, second pass, third pass escalating up in friction. Now on the right, now this is looking at proteoglycan 4, and because there's another variable in here of the dose dependency, they're choosing the fourth position on the left, so that fourth sort of wipe of that eyelid you can think of, and taking that data and putting that on those plots, but now looking at the various dosage level of this proteoglycan 4. So you can see the higher dosage level with significantly less static friction. So you can see lots of variables here, lots of ways to plot it, and hopefully that comes across and is clear. Okay, so here we see a final set of plots. Again, static friction, kinetic friction against sliding velocity. And here we have an addition of this AQ parameter AQ plot is an off-the-shelf hyaluronic acid. And what we see is when you combine proteoglycan 4 with that solution, you get not only the benefit from proteoglycan 4, which is quite a bit less, but on the kinetic friction, even lower friction still. So that in combination looks like a very powerful advantage, especially when you look at relative to saline solution or this standard hyaluronic acid solution, something like half the friction values in these plots. So in the final section now, we're going to talk about tissue stimulation. And this is using a variation of the products that we've been talking about, but now with some specific application in growing cells or driving tissues that have active cells, live cells, and driving differentiation and allowing reorganization and regeneration of those tissues to be more appropriate for future implantation as substitutes for medical devices made of metals and polymers. So we're going to touch on a couple studies here. The first study comes out of the University of Sheffield, and it's looking at using human mesenchymal cells, and they're not as potent as stem cells at differentiation. And one of the tactics to overcome that is to use pharmacological solutions or drug solutions to generate cell differentiation, but those have side effects. So here the purpose of this study is to investigate, can you use dynamic compressive loading on these cells to then drive differentiation, and can that then be an alternative for creating these tissue engineered medical constructs. So here, the instrument we're using looks familiar. It's a 3200 that we've seen used in lots of characterization studies so far, but now it has what we call a biodynamic chamber. That's just a fancy name for our bioreactor chamber that allows you to, in a sterile environment, sterilize all the chamber components and install your samples, your tissues inside of them. And then in this case, sort of clip that into the 3200 and allow the 3200 instrument to apply axial compression, still measure force, and using that loading, you're essentially stimulating the tissue in this biologic environment, and you can flow fluids through it and control the biological environment. but look for then the variation and the changes in those mechanical properties during that growth cycle. So on the left, we can see the test method employed, where we're applying 5% strain at 1 Hertz, but really only 2 hours, occasionally in this period through a 24-day study. And you can see in the timeline down below where they're adding various markers, and in some cases the dexamethasone. So the dexamethasone was a variable here that's essentially the steroid alternate that they're looking at. Is there a viable alternative for this, because that steroid has some negative effects. So on the right side, we see a first round of results where the ALP, this is the alkaline phosphatase activity, and that increases during bone resorption. So the fact that that is high in the first 3 columns, you can look at the first column is applying mechanical stimulus plus dexamethasone, and then the second is without loading stimulus but with dex. The third column is with load but without the steroid, and then the last is with neither forcing stimulus or the steroid. And you can see that there's significant difference there, where the first 3 is much different than the last. So that's our first clue that we're getting good bone resorption, and we're differentiating them into this proper cell lineage with really any 3 of those combinations on the left. So when the scaffolds are stained for collagen and calcium mineralization, we get to visualize what's happening here, and we see a similar pattern. So here, dex can also contribute to the collagen formation as well as the calcium mineralization, but so does the loading. So but there isn't a synergistic behavior by employing both of them together. So this gives us a clue that, okay, doesn't really help to do both. You can really use one or the other. So it concludes that this harmful effects of steroids may be viably removed, and the alternative could be mechanical stimulation in the generation of this tissue. In this final study we'll review today, we're looking at smooth muscle cells that are obtained from naive rats, and this study is from McGill University. So smooth muscle cells, they're part of the vascular structures found in the human body. And the study shows at a high level that the importance of physiologically relevant forces that's important to create proper cell proliferation. It's not just about simple tension or compression. In this case, we're going to be looking at an airway, and that's a radial, sort of a tube-like structure, and we'll see that actually radial distension and shear forces in that in the inside diameter both play a critical role in creating or proliferating and differentiating these cells to be more effective airway substitutes. And so here we're going to see a new instrument on the left. It's a biodynamic instrument. This one's kind of a simpler version of many of the other ones, which is really just to create pulsatile flow through a vessel. In this case, it's an airway, so it's tube-like. And then on the right, we're actually also using a 3200 in the same lab, where they're using that for measuring mechanical properties after it has been engineered and conditioned in the biodynamic system. So the point here is to understand this effect of shear stress, circumferential strain, on how these smooth muscle cells are reacting in a tissue engineered airway. Okay, so in the test method, so now we see a few other parameters here. We see milliliters per minute. This is like a flow rate of the fluid through a tube, and now we can actually oscillate that flow rate. So this is kind of like your heart does. It pushes fluid and blood through your vessel and then effectively slows down or stops and then pushes it again. So this biodynamic system allows us to do this by sort of pumping fluid back and forth. We use this pulsatile manifold on the right. We combine it with a gear pump, and this allows us to pulse fluid at a controlled rate through this tubular structure. And you can see some illustration here of how they're creating this, originally a sheet, cellular collagen, casting that and then rolling that up around a mandrel, and that's what's creating this, this tissue engineered airway. So now let's look at some results. On the left, we see a plot of the number of cells within that scaffold structure over time, and comparing specifically the approach where you apply a mean flow, sort of a constant shear flow and fluid flow through the center of this of the airway. And then the darker bars are when you're pulsing fluid. So your shear stress and the pressure are both oscillating and increasing and decreasing in a cyclic way throughout that stimulation. So we see a significant difference there in the number of cells created, proliferated. On the right, you see some nice images of stain material where now you can see some how these layers, if you remember, there's a sheet and they're rolled up essentially to create a tube. And now you can see on the left there's detachment and sort of not a very cohesive structure across that wall thickness, where the pulsatile cross-section looks much better and much more like a single tissue there. And here are some nice photographs from the scanning electron microscope and confocal laser scanning microscope. On the left, you'll see images from the inside of the vascular scaffold. On the right side, you see images from the outside of the vascular scaffold. So as we look from top to bottom, the cells are arranged in essentially a random way where there's no meaningfully flow. So it's a static. This is a airway that's just in the biodynamic chamber, but doesn't actually have any flow at all. So the middle one is a mean flow, just a laminar flow through the middle, and then the bottom row is when we're actually pulsing them. And what we see here is what we're looking for, this alignment of cells and this tissue, where interestingly on the bottom with the pulsatile flow, we see cells on the inside arrange themselves in the flow direction, but cells on the outside arrange themselves in the direction of pulsatile stretching. So that's perpendicular to flow, kind of a hoop stress, response to the hoop stress of that tissue. So this is kind of wrapped up here in this slide with a little bit of an illustration here, where you can see the biomarkers and mechanical properties. They're decreasing with static fluid, and there's some change, minor change with the laminar sort of static fluid flow, but we see significant improvement in the biomarkers and the alignment of tissue, and then the mechanical properties of the final tissue when tested, when you have this superposition of pulsing fluid stress as well as pressure in this tube that's creating then radial stress, and that tissue then sees that and essentially is stimulated by that or learns from that and adapts. So this shows the sort of incremental value of different types of mechanical stimulus, and in this case, because the airway is going to have different stress types on the inside versus the outside, it's important to replicate some of these paradigms of stress, you might say, to create better tissues. Okay, with that, I will wrap up and just kind of pause here. And what I try to do today is give you a few examples in a few different areas of tissue or biomaterials, and in some cases tissue engineered products. But we work with our customers and researchers out there they're working on all sorts of different components of the body. Again, sometimes medical devices, sometimes just the biomaterial characterization. And it's just really encouraging to see the progress made and And it's encouraging when we have instruments that significantly enable that. That's really what we focus on. And hopefully this is helpful today for you to appreciate some of the work that's out there and some of the things you can do in different ways. You can design methods, you can execute tests to enable your research. So thank you, everybody. Thank you for your time today.
Thank you, Troy. This series will be talking to many of these material properties, how it can help you measure, and how it can help you understand your material better. Next week's talk will be on fatigue and durability of medical devices, presented by Mohammed Harris. We hope to see you then. We'll now take some time for questions. Whilst we collate the questions that you've been submitting during the talk, we'd appreciate if you could answer a short poll to help us prioritize how we can help you better. Thank you. Okay, thank you for answering the poll. So for the questions, we are being joined by next week's presenter, Mohammed Harris. Harris is an application engineer for Electroforce covering Europe. He specializes in Electroforce mechanical test instruments and engages with customers on their testing needs. He joins Troy as our experts to answer your questions today. So we do have a little bit of time to answer some questions. So, first one, can you explain a bit more about the environment? I think you said a bath and then a biodynamic chamber. What is the difference?
Thanks, Ben. Yeah, that's a good question. Didn't talk too much about what we would call a standard bath, but I did reference biodynamic chambers. And essentially the difference is in a standard saline bath, those are typically used in a non-sterile environment, typical laboratory environment. They're good for testing of materials in saline, phosphate buffered saline, deionized water, things like that, but they're not meant to grow tissue or sustain life in tissue, more just to create the right environment of temperature and look at some corrosion studies in medical devices or materials for that. The biodynamic chambers and then the instruments they sort of clip into and are stimulated with are all designed to go into a cell culture incubator. First of all, they're designed to go through autoclave, get unwrapped in a flow hood, assembled in its sort of entirety in creating a flow loop, closing the flow loop, then transporting that quote-unquote biodynamic chamber over into the cell culture incubator, clipped into the instrument that's waiting for it, close up the incubator, the flow loop stays closed there. There's a media filter in the flow loop, and then close the incubator door, and then you start your longer-term study. So the biodynamic chamber, sort of a bioreactor chamber in this stimulus instrument is what we would call then a mechanical stimulation bioreactor. So that's the biodynamic tissue growth versus a more basic saline chamber. Hopefully that answers the question.
Excellent, thank you. Another question is around, so a practical question. Can I pause my test for inspection and resume the testing?
Yep, that's a good question, especially in these long-term studies. You know, if you've got multiple days, whether it's a fatigue study or whether it's a tissue growth study, you can do it a couple ways. In the software, you can always manually click sort of pause or hold or stop the test, and then inspect. Sometimes it's taking out the chamber and putting it into some sort of microscopy system, or just opening the door and looking at it. In other cases, it could be removing the sample, inspecting it, reimplanting the sample, and then resuming in the software or starting that test kind of where you left off. So you can do that manually, or you can really script that. You can say, and we saw this in some of the studies where there was partial loading, just I think, what was it, 2 minutes a day or something like that, where you can script to say you can load for this amount of time, then you can hold for some period of time, and in that holding time you could inspect your sample. So you can manually interact with the instrument, stop, or you can script that sequence.
Okay, and in a very similar vein, there's a follow-up question to, so when you basically, can you change the loading levels after the test has started, or do you need to actually stop and restart?
Yep, that's a good question, and that's actually the same answer. You can do that in the software manually at any time and say, hey, I was running from -1 Newton to -10 newtons. This sample, I feel like now I've seen something to say I want to run to -20 newtons. So you can just go in and you can kind of edit that motion on the fly. It won't even stop. It'll just run, maybe it's 1,026 cycles that you've done, and then on the 1,027th, you click 20 newtons, then the next one, it will load to 20 newtons. Or, like we said before, you can script that. You can say, I'm going to run 1,000 cycles, then 2,000 cycles, then 3,000 cycles, and those could be all at different levels, different frequencies. You could switch from load, sort of force control, to displacement control. You can put a pause in there. All those things can be scripted. That's quite popular. That's what we see in these stimulation studies. Is very seldom do you stimulate it 100% of the time. You're often stimulating it and then letting that tissue rest and remodel at that point.
Okay, that's prompted another question along similar lines. So how long is a typical test on tissues?
I think one of the studies we saw was 30 days. That's kind of, I would say, on the longer study. 1 week is kind of a common. But if it's a tissue, not a tissue growth study, it could be 2 minutes. So you're just looking at compression tests. But when we're growing tissue, looking for remodeling, cell differentiation, driving that, then we're looking at something definitely more than a day. And that's where the sterile environment's important. Following good practices in assembling these chambers in flow hoods, putting it in incubator, cleaning the incubators. Those are all good practices to help those long-term studies.
Okay, so just a question here. So I have a question here around stents. The stents made up of a nickel titanium alloys implanted into a patient. Will there be any possibility of stress on stents due to the flow of blood, the amplitude? Will it weaken the strength of the test?
Yeah, that's a good question. I don't know that I can definitively say yes or no, but I think in most of what we see is the failure modes of whether it's stainless steel or nickel titanium is predominantly a mechanical stress created, in this case, due to the radial distension due to the change in pressure of that vessel. That's almost always the more significant stressor. Yes, technically if the stent is exposed on the inside of the tube, there's some longitudinal stress and shear stress on that material, but that stress level is, I think if you analyze that, will be orders of magnitude, maybe at least one order of magnitude less than the oscillating stress. And I think that's consistent with the failure modes you see clinically, and therefore much of the standards that you see are written around that. I will say there's maybe one exception in that if you have a stented heart valve, or maybe a filter or something that has the need to resist axial motion down the vessel, or in the heart valve, placement area where the valve is closed, you have pressure that's exerted on the leaflets. The pressure times the area is going to create that force, and then you've got force that wants to migrate that device, that nickel titanium stent in this question, down it, and you might have anchors, or in a vena cava filter you might have a blood clot, and that blood clot has shear flow, and then that creates axial force. So I think the point is you have to always look at the physiologic loading case by case, and stents, because they're just so successful out there in the minimally invasive therapies, that they're getting used more and more in lots of other areas, not just to hold the coronary vessel open, as they did 20 years ago. So you'll always have to look at it. I think if you're just looking at a stent, the shear force is relatively low.
Excellent, thank you. Well, we have actually, just noticed, John, just gone over time, so I'm going to draw this to a close there. Any questions we haven't answered, we'll endeavour to do that over during this week. So thank you very much for attending. Thank you, Troy, for the presentation. And I hope to speak to you all again next week for the next instalment. Thank you very much.
Thanks, Ben. Thanks, everybody.